Hello everyone. Let's get started. Today we're going to be talking about the history of microbiology and the science of microscopy. Humans have a long and twisting history of our understanding of the origin of diseases, particularly infectious disease. Originally, as far back as the ancient Greeks and ancient Romans, people primarily believed that disease came from the gods. When humans misbehaved or failed to follow the rules of their religion, that they were somehow punished by the gods in the form of disease and other disasters. It wasn't until the time of Hippocrates, back between 4 and 500 BC, that people started to think about disease a little bit differently. Hippocrates, of course, is the man that we credit with being the father of Western medicine, and he proposed that disease, rather than coming from the anger of the gods, instead arose because of a problem within the human body itself. Hippocrates believed that there were 4 humors in the body, and that when these humors went out of balance, that brought upon disease. This idea was influenced by some of the beliefs in Eastern medicine. Now, if you're not familiar with the 4 humors that Hippocrates wrote about, they include the blood, the phlegm, what he referred to as black bile, and also yellow bile. And Hippocrates would have considered anything that sort of exited the body up and out of the mouth as being a bile substance. Now, following Hippocrates, in the 300s BC, Aristotle wrote about a different idea around the origins of human disease, and more specifically around the origins of infectious disease. Aristotle is the father of what we call the theory of spontaneous generation. The theory of spontaneous generation states that living things can generate spontaneously out of non-living things. So, for example, Aristotle was referring to what seemed like the spontaneous generation of insects out of dust and dirt, the generation of maggots spontaneously out of rotting flesh, and even what appeared to be the spontaneous generation of rodents inside stored grain piles. It wasn't until about the 100s BC, when a man named Marcus Varro started to write his ideas, that people began imagining that there might be organisms on the planet that were too small to see, that there might be these little creatures that were involved in disease, particularly infectious disease, that were simply invisible to the naked eye. Barrow also wrote about a belief that disease was associated with the environment. So he began to speculate about the role of bad air in the development of disease. He noted that places like swampy areas and natural locations that tended to smell very bad from gases emanating from decaying material, places in cities that were particularly filthy, that were sometimes overlaid with garbage and human excrement and things like that, that these places that had a terrible smell to them, that appeared to be quite dirty and filthy, they created this bad air, and somehow being exposed to this bad air as a human caused the risk of developing disease. Now, throughout the ages, really up into the mid part of the 19th century, the theory of spontaneous generation really persisted. A lot of scientists held on to that idea that living things could actually generate out of inanimate objects. But competing theories began to arise in the 1800s. One of those theories is referred to as the Miasma Theory. Now, the Miasma Theory also centers around this concept of bad air that Varro had introduced. That it's bad air, this dirty, filthy, somehow contaminated environment, and the air that emanates from those places that causes disease, that filth was dangerous, and unsanitary conditions, decay, and so on. These things were dangerous to humans because they could bring about disease. If you think about it, miasma theory was heading in the right direction because, as we know, places that are particularly filthy can also be absolutely ripe with microorganisms, and some of those may be pathogens. But miasma theory did not take into account the idea that infectious disease could somehow be contagious from person to person. Instead, the miasma theory stated that anybody who was exposed to the bad air was susceptible to disease. But one human with disease didn't spread it to other humans. That was not part of the theory. So there was no concept of contagionism in miasma theory. Now, during the years, the 1600s, the 1700s, and into the 1800s, while miasma theory was sort of competing with the theory of spontaneous generation and other new emerging theories, this new science was being born. This new field of life science was being born, and that was the field of microbiology. A scientist named Robert Hooke is credited with developing the very first, and very primitive by today's standard, microscope. Hooke created this magnifying device, and he very famously examined a piece of bark from a cork tree. And under his microscope, he could see these little compartments inside the cork bark. And it was Hooke who described these little compartments as cells. We still use that term cell today to describe essentially the compartment of life, the unit of living things. But Hooke would not have had that understanding. Instead, that word cell that he used to describe these compartments, that word came from monasteries, which were very, very popular in Hooke's day. Monasteries, of course, are where monks and priests and religious people would live. They would live a very sparse existence, and they would each be assigned a very bare compartment to sleep in, and that was called their cell. Inside a man's cell would be, a very simple bed and maybe a chair, maybe a small table, but that was it. And this was their cell. This was the place where they went to rest, to sleep. So Hooke used that word because when he saw those little compartments under the microscope, it brought to mind these rooms inside of a monastery. Now, a contemporary of Mr. Hooke was another scientist whose last name is Leeuwenhoek. Antony van Leeuwenhoek. It's a terrible word to spell, as you can see. Leeuwenhoek developed his own primitive microscope to examine living things. And he actually described what he called a tiny animal underneath his scope. So he was looking at materials where he could see microbes that were capable of movement. Very famously, Leeuwenhoek took his own semen and placed it on a slide underneath his microscope. And of course, sperm cells are capable of movement. And so he came up with a term, animalcule, to describe these living things that were too small to be seen with the naked eye that existed in nature. Now, these again would be very primitive microscopes by today's standards, but they really were the first of many generations of microscopic technology. In the upper left-hand part of this slide, you can see a painting of Robert Hooke and a drawing of the microscope that he developed and used to examine cork bark. And over here on the right-hand side, you can see an image from an old text, which was a drawing of these pieces of cork bark that had these little compartments in them, these little cells, as Hooke called them. Leeuwenhoek is shown on the lower left-hand side, and Leeuwenhoek's microscope would have been based somewhat on the design of Hooke's scope, but it was a little bit more advanced. He was able to see small objects with a little bit more magnification, but again, nothing compared to today's technology. So the theory of spontaneous generation and miasma theory sort of sat side by side for several centuries until in the early and mid 19th century, a couple of other theories started to gain popularity. One was called contingent contagionism, and this was the first of these theories to introduce the concept of disease spreading from person to person. That if a person became sick with a certain type of disease, an infectious disease, that they could then pass that disease on to another person. This was a really important step forward in our understanding of infectious disease. However, there was still no understanding of where that infectious disease came from, how that first person became ill. There was still an idea that it was exposure to an unhealthy environment that brought about the disease. Zymotic theory was a little bit different from contingent contagionism. Zymotic theory was the idea that there were certain diseases that were different from others. And of course, these would be the infectious diseases that we know of now. Diseases that caused fevers, for example. Diseases that could be spread, again, from one person to another, were due to what they referred to as a fermentation process going on in the body. Now, they were really headed in the right direction with zymotic theory because, of course, we now know that it's microbial organisms that ferment. But zymotic theory occurred at a time when we didn't really have an understanding of why fermentation occurred, just that it did. So by describing disease, infectious disease, as being the result of some kind of fermentation in the body, again, they were headed in the right direction, but they had still not associated the microbial organism with the development of the contagious disease.